Hi everyone, welcome to this GCSE Biology revision webinar. This one is going to be on the subject of organisation. So we've just completed our sound check and everybody that I've expected, or almost all of the students from the 4 schools and also the private students, almost all of you have now logged in and confirmed that the audio and video is working okay for you. If that does change, if there are any audio-visual problems during this webinar, please do let me know through the chat box on your interface, whether you're using a browser or whether you're using the Zoom application. That way I can take corrective measures, but please do just reserve the chat box for messages like that. If you have any questions, as always, during this webinar, please do type those not in the chat box, but in the question and answer box on your interface. That way other students in your Zoom webinar room will be able to see your question and upvote that question if they feel like they'd also like that question to be answered. And again, as with the previous webinars in this series, I will be going through those in the Q&A section at the end of this webinar in the order in which they received votes. So in the first hour, as always, we're going to be going through some of the topic teaching, again in the order in which you voted for the topics. These are the topics that we are going to be attempting to cover, not necessarily in this order. We're going to go through them in the order, as I say, in which they received votes on the private webinar resources page. And then we'll have a 5-minute break at the end of that first hour, after which we'll continue the teaching. Now, during the 5-minute break, or towards the end of the first 5-minute break, you will see a poll pop up on your screen where you will be able to vote as to whether you'd like me to continue with the exam questions that you all received as part of your ticket booking process for this webinar. And if that is the case, if that receives the most votes, then we'll be going through these exam questions again in the order in which you voted for them. Alternatively, you'll be able to vote as to whether or not you'd like me to continue with the topic teaching for that second hour. After that, we will have the option to extend the webinar for an extra 30 minutes. Again, a poll will pop up on your screen, and whether or not you choose for that extension, you'll be able to vote again as to whether you'd like me to continue or extend the webinar by 30 minutes on the topic teaching or on the exam questions. And then finally, in the last 30 minutes, we are going to be going through the Q&A. So if you do have any questions after the webinar concludes and you'd like to ask me anything, please do go to the private webinar resources page, where you'll be able to find all the resources for this webinar uploaded roughly within 48 hours of the close of this event. And there you will also be able to find the live chat, where you'll be able to ask questions and discuss things with other students and myself. You'll also have the ability to send private messages via that page to me, which I will then answer privately. So with that housekeeping out the way, let's get into the topic for today, which is organisation in biology. Before I forget, actually, if you ever do need to get hold of the link for the private webinar resources page, just have a look at the first page of your version of this document, which will be a PDF that you downloaded with your ticket. And the second link on that page is the link that takes you to the private resources page for this webinar. All right, so we've got a lot to get through. This is a very large topic, regardless of which specification you happen to be studying for. So, organisation in biology then. In the first GCSE Biology webinar of this series earlier on this week, we covered the topic of cells, specifically cell biology. We began with the definition of cells and looked at cell differentiation and cell specialization. Now, those ideas underpin this topic, so I'm not going to be going back through those. Those of you who attended those webinars will be able to watch the replay video for that one, but just bear in mind the idea of a cell, the definition of a cell, underpins the ideas that we're going to be talking about here. But this topic, for most of you, is not about individual cells per se. It's about the ways in which those cells are organized into tissues and organ systems and so on. So in biology, we are dealing, once again, as I said before in that first webinar, with extremely complicated systems. And so as scientists, it's useful for us to break them down and organize those systems into levels of complexity, levels of organization. Now, to some extent, this is an arbitrary categorization of these systems in order to make it easier for us to teach and learn about them, and also easier for us to communicate about them as scientists. But nonetheless, as a biologist, you do need to know about the hierarchical levels of organization within biology. So these are as follows. The first is the cell, which is the smallest living unit of matter, which performs all of these functions which are characteristic of living things. So movement, respiration, sensitivity, growth, reproduction, excretion, nutrition, and some people would also argue that homeostasis is a characteristic of living organisms. Now, if you are dealing with a multicellular organism, then those cells can differentiate into specialized cells to perform specific functions within that multicellular organism. Specialized cells can then be collected together, specialized cells of a similar type, grouping together to perform an overall set of functions within that organism is referred to as a tissue. Now, different tissues can group together to form an organ, which is a structure that carries out one or more definite functions within that multicellular organism. And we're going to be looking at some examples in a moment of all of these, including organs. And finally, multiple organs can work together within an organism in various ways to form what we call an organ system. And once again, we're going to be looking at some examples in both animals and plants. Now, all of these different tissues, organs, organ systems work together within a multicellular organism that can carry out all those 7 or 8 characteristics of living things. So this is really the nuts and bolts, the foundations of what we're going to be talking about. So let's start by looking at animals. Animal tissues first, and then we'll move on to plant tissues and organs, because that is the order in which you voted for them. Now, specifically, those of you who did place votes on the resources page, you requested that I go through the circulatory system first, followed by the digestive system. So we are going to be doing that. But before we go into these organ systems and specific organs and their structures and functions, you do need to know a little bit about animal tissues and the ways in which those are categorized functionally. So there are 6 principal kinds of human tissues. There is epithelial tissue, which is the tissue which tends to form the lining or the surfaces of structures in the body. There is connective tissue, there is skeletal tissue, there is blood tissue, there is muscle tissue, and there is nervous tissue. So this is the kind of categorization that most biologists will use to describe the different kinds of tissues, and they're categorized by their function. But again, in biology, you're probably sick of hearing the phrase structure versus function, because it is the whole basis of understanding biology. Structures in biology, whether you're talking about molecules like proteins and nucleic acids, or you're talking about organs and organ systems and whole organisms, the things that those structures do, their functions, is largely determined by their structures. So to remind you, all of these are tissues, and so they all match this definition of a tissue, a group of similar specialized cells which work together to perform a particular function within the organism. So if we take a look at some of the human tissues, in fact, you'll find that these are the same kinds of tissues which are also found in many kinds of animals as well. So, to summarize what we've been saying so far, a tissue is made up of specialized cells. So here's an example. Nerve tissue, which you find in the spinal cord and in the brain, for example, it's made up of specialized, long, thin cells whose kind of specialization allows them to transmit nerve impulses, electrochemical impulses around the body as signals which can control movement and also which are responsible for sensation. So these impulses can travel very long distances in terms of cellular distances. So cells are generally measured on the order of micrometers, as we saw in the previous webinar on cell biology. But these cells, they may be very small in terms of the size of their cell body or their width, but they are extremely long. So certain motor neurons, for example, go all the way from the motor cortex, which is in the top of the brain, and their axons, these long, thin structures, run all the way down to, for example, the muscles of your big toe. And so these can obviously there are synapses between nerve cells as well, but nerve cells, in terms of their length, are very, very long for a cell, which itself is such a small structure in general. So this is the general structure of a nerve cell. We see here the cell body with the dendrites and the synapses between the dendrites of this nerve cell, this neuron, and other neurons that you can see here synapsing onto its cell body and onto its dendrites. We see the axon hillock from which emerges this long axon. So this is the cell body of, for example, a motor neuron, but this is the typical structure of a sensory neuron. You have a sensory nerve ending, for example, a nerve ending which is responsive to heat or pressure or pain. You have a long axon which transmits that information as a series of electrochemical impulses to the central nervous system, where it synapses onto other nerve cells to process that information. And here you can see the cell body of this neuron. So you can see that the structure of a nerve cell is extremely highly specialized, whether you're talking about a motor neuron or a sensory neuron. This is a photograph of nerve cells, neurons, as seen with an electron microscope, and so you can see some of these features in this photograph. So the nerve tissue, carries impulses that themselves are responsible for either causing effects like muscle contraction and the secretion of certain things from glands, or it can also be going in the opposite direction for transmitting sensation from the periphery of the body to the central nervous system for processing. So we've talked about nerve tissue, but nerve tissue, muscle tissue, and bone tissue work together in the musculoskeletal system to allow us to move. So here we can see nervous tissue in the brain, which works with skeletal muscle tissue, which itself is attached to bone tissue via connective tissue like tendons, and the bones themselves are connected via another form of connective tissue called ligaments. So nerve cells, neurons, and muscle cells will form junctions between them, neuromuscular junctions. So here we can see how different types of tissue work together in systems. So here we can see again the neuronal cell body. This is a motor neuron with its nucleus here. You can see the nerve fibers, the dendrites, and you can see here the motor end plate, which is where you have a junction between the ends of the motor neuron and the muscle cell membrane. And it's here where the signal gets transmitted from the neuron to the muscle cell, and the muscle cell then contracts. Here we can see the axon, the nerve fiber, the extended long thin nerve axon with the myelin sheath. So nerve, muscle, and bone tissue work together to allow movement. So all we can do and think is initiated, carried out, and controlled by specialized cells all working together in tissues. So a system, once again, is made up of several organs and also tissues which work together to perform specific related functions within that organism. So here are some examples here, and we're going to be looking at some of these, in particular the circulatory system and the digestive system. Here are some more examples in the body. Okay. All right, so let's start by looking at the specific organ system that you all voted for, or rather that received most of your votes, which is the human circulatory system. So the circulatory system is sometimes called the cardiovascular system, and it's composed of blood, which is a tissue. It's also composed of blood vessels, and it's also contains a single organ, the heart. So there are three main types of blood vessels: arteries, veins, and capillaries. So we need to look at the structures and the functions of all of these and how they're related to each other. So this is a map of some of the main arteries in the body, but arteries will subdivide into finer and finer arterioles, which eventually will end up as dividing up into very fine vessels called capillaries, which are networked together in what we call capillary beds. And then those capillaries join up again to form venules, which are very, very fine, very thin veins, which join up to form thicker and thicker veins until you get very large veins like the great saphenous vein of the legs, joining together to form the vena cava. the vena cava, the superior and inferior vena cava, which carry blood back to the heart. So this is a kind of overview of the circulatory system. So let's take a look at the major blood vessels. Now this is a highly simplified view. So we have the pulmonary artery, which is the only artery which carries deoxygenated blood. So this carries blood from the right ventricle of the heart to the lungs, and then back again. It splits into the left and right pulmonary arteries. Here we can see the right atrium of the heart, the right ventricle. So the vessel which carries blood back to the heart is called the vena cava. Here we can see the vena cava. We can also see here that there's a vessel which carries blood from the liver back to the vena cava. This is the hepatic vein. Here we can see the hepatic portal vein, which carries the blood containing the products of digestion from the small intestine to the liver. Here we can see the renal veins. So the renal veins are left and right. Again, this is a simplified diagram. The left and right renal veins actually join the inferior vena cava, which carries blood back up to the right-hand side of the heart. So here we can see the aorta, which is the largest main artery. This emerges directly from the left ventricle of the heart, which is where we have the blood being pumped out, and this contains oxygenated blood, which has just been received from the left atrium, and the left atrium receives that oxygenated blood from the lungs as it flows back from the lungs, where it has exchanged the carbon dioxide for oxygen. So oxygen is dissolved into the blood and carbon dioxide is diffused out. This is oxygenated blood here, which is coloured red in this diagram. Deoxygenated blood is coloured blue. Now, we also have the lungs themselves, obviously. So this would be the hepatic artery. So the hepatic artery is the artery that carries oxygenated blood to the tissues of the liver. And here we can see the small intestine, and the small intestine is supplied by the mesenteric artery. So here we can see the renal arteries, the right and the left renal arteries here. So this is a kind of overview of the main arteries of the body, but there are many, many more, and this is a highly simplified diagram. So for example, the aorta also gives off branches to the head and the neck, which are the left and right carotid arteries. It gives off branches to the arms, which are called the subclavian arteries. As it descends the abdomen, it gives off branches, left and right branches to the legs, the iliac arteries, and so on. But these are the main arteries that you need to be aware of at GCSE level. So what are the main differences between arteries, veins and capillaries? Well, this diagram here illustrates some of those differences. So let's start with a diagram of an artery here. So an artery has thick walls made up of muscular and elastic tissue. And it also has a sheath of connective tissue around it. So arteries carry blood away from the heart, generally speaking. So this is a good generalization. Most arteries carry oxygenated blood, with the exception of the pulmonary artery. Now, the reason they have such thick muscular walls is because they have to withstand the pulse pressure, the hydrostatic pressure generated by the squeezing, the contraction of the left ventricle, which is very, very powerful. So, the veins, in contrast, typically have a thin wall made up of thinner layers of smooth muscle and elastic tissue and non-elastic fibers. So the veins are distinct from the arteries because they tend to carry blood in a direction towards the heart. Now, typically, veins carry deoxygenated blood, and the only exception to this are the pulmonary veins, which carry oxygenated blood from the lungs towards the heart. Now, capillaries are extremely thin, microscopic blood vessels which have walls which are 1 cell thick. So this is a light or optical micrograph of a bed of capillaries here. So these are extremely important for the exchange of nutrients and oxygen and other substances from the blood to the surrounding tissues through these thin walls of the capillaries. So it's in a capillary bed where the exchange of nutrients, waste chemicals, respiratory gases, and other substances take place. Certain substances go into the capillaries from the surrounding extracellular fluid. Certain substances will diffuse out of the capillaries into the extracellular fluid. So it's also via these capillary beds that arterial vessels eventually will merge into the venous vessels. So the sequence, as I said before, is this: the arteries divide. So if you imagine this is an artery here, it divides into successively finer arterioles. Eventually, so here we have an artery, successively finer arterioles, which eventually divide into capillaries, which form these networks called capillary beds, which surround the cells of the body tissues. Those capillaries then join back up into fine, very thin venules, which then eventually join together to form veins. So tissue fluid, the fluid which bathes the outside surfaces of these tissue cells, is made up of the blood plasma minus the large plasma proteins and blood cells, which forces itself through the walls of the capillaries and forms the tissue fluid. So tissue fluid eventually would build up if it wasn't drained away, and that's one of the functions of the lymphatic system. So these are lymphatic vessels whose function it is to drain away the excess tissue fluid into the lymphatic system, where eventually it returns to the circulatory system. So the fluid in the lymphatic system is called lymph, but it's essentially just the extracellular fluid which has been reabsorbed into the lymphatic vessels so that it can be returned eventually to the circulatory system. Otherwise, your tissues would simply swell up with accumulating extracellular fluid as the fluid leaks out of all of these numerous capillary beds into the extracellular medium. So, sorry, this is slightly off center here. This is the lymphatic vessel. These are the capillary beds here, and this is the tissue fluid. And these arrows represent the movement of the tissue fluid out of the capillaries. So capillaries are usually found, as I said before, in capillary beds. This is a slightly more magnified view of that same micrograph here. Now, in fact, capillaries are so numerous that every single one of your cells of your body are very, very close to, within 1 or 2 cell widths of a capillary. And this gives you a large amount of capillaries suffusing the entire volume of your tissues. And the reason for this is that the main way in which substances such as oxygen and nutrients diffuse out of the capillaries is by diffusion. And diffusion only really works effectively over very, very short diffusion distances. Now, we talked about this towards the end of the last webinar on cell biology, where we spoke about diffusion and so on, active transport and osmosis and so on. Diffusion is only really efficient over very short distances, and so if every single one of the cells of your tissues of your body require a constant supply of oxygen, they require a constant supply of glucose and other nutrients, they also require the constant removal of carbon dioxide that builds up inside them due to respiration, well, it means that all of those substances, if they can only be transferred by diffusion, you need to have a capillary very, very close by to those each of those cells. Otherwise, those cells are going to die from lack of oxygen or excess metabolic waste, such as carbon dioxide building up. So every individual cell of every individual tissue of every organ is very, very close to a capillary, and this gives you a large amount of surface area and also ability to deliver substances and exchange substances between the blood and the tissues. So here we see a image, well, rather a computer-generated image of what blood cells may look like inside a capillary. Now, in fact, in capillaries are only just wide enough to allow blood cells, red blood cells, to move through them in kind of single file. And so this gives you a sense of just how small capillaries are, the diameter of capillaries. So to compare arteries and veins, here is a transverse section. This photomicrograph shows a small artery and a small vein. You can see the difference in the thickness of the walls of the artery versus the vein here, and also the larger diameter, the larger lumen of the vein versus the artery. So here we can see another micrograph of the transverse section through an artery. Now, the word lumen means the inner compartment of something, so the inside of a compartment, like an artery or like a organ. So here we can see connective tissue. We can also see the smooth muscle and also elastic tissue, which makes up the thick, much of the thick walls of an artery. Here we can see a type of epithelium called endothelium. So epithelium, generally speaking, is a tissue made up of a thin layer of cells, which is designed to form the lining of the compartments of the body and also the surfaces of structures of the body. That's the general term, but epithelium properly applied is applied to the surfaces of objects. So for example, your skin is made up of layers of stratified epithelium, and that forms the outer covering of your body. But equally, the inner surfaces of your body compartments are made of epithelium as well, but in that case it's called endothelium because the prefix endo means within. So the blood pressure is lower in veins than in the arteries, and this means that unlike the arteries, the veins do not have the hydrostatic pressure of the pumping of the heart causing the blood to move through them. The veins rely on a different mechanism to ensure that blood flows in a specific direction. Veins have labels in them. So if we imagine that this is one of the veins in your leg, the blood needs to be returned against the force of gravity, and because there's no hydrostatic pressure of the heartbeat in the veins, something else needs to provide the pressure, but also you also need some way of preventing the blood from slipping downwards under the influence of gravity and pooling in the legs. And so this is provided by the contractions of the muscles around the veins. So what you have is muscles around the veins, which when you move, those muscles contract, those squeeze the veins. This causes the motion of blood through the veins. The valves will open because the valves ensure blood flow only in the correct direction. Now, when the muscles relax, the valves will close as the blood starts to slip back, and this causes the blood to always flow towards the heart in veins. So let's take a look at the human heart then. So the human heart is somewhat typical in its structure in terms of comparing it to other the structures of the heart of other mammals. So it's an organ that pumps blood around the body, and it's found in a section of the body called the thorax. Now the thorax is essentially your chest cavity. It's everything from the diaphragm all the way up to the rib cage. So everything between the rib cage and the diaphragm is what we call, this section of the body is what we call the thorax. So the heart is situated in a region of the thorax called the mediastinum. And so it's just beneath the lungs, as you see here, and it is protected by the rib cage, which we're not seeing because this is a cutaway model here. So there's the heart, and the heart does have 4 chambers. So here we can see a diagram of the pumping action of the heart and those 4 chambers. Let's just go ahead and label those. So I referred to some of these chambers in the previous slides here, but we're going to be looking at this in a little bit more detail now. So let's take a look at the actual internal structure of the heart in a little bit more detail here. So here's an anatomical diagram here. We can see the superior vena cava. This is the vena cava which carries deoxygenated blood back to the right-hand side of the heart from the head and the neck. And so this is entering the right ventricle of the heart here. This is one of the 2 pulmonary arteries. So the pulmonary arteries carry deoxygenated blood from the right ventricle, which when the right ventricle contracts, it squeezes blood through the left and the right and the left pulmonary arteries. Now you'll notice that in these anatomical diagrams, your left, as you face the diagram, is in fact the right-hand side of that heart, because by convention, anatomical diagrams are drawn as though you're looking at the organ of a patient who is sitting facing you. So if this is the heart in the chest of an organ of a patient who is facing you, their right is your left, their left is your right. So this is the right-hand side of the heart, the right atrium, right ventricle. This is the left-hand side of the heart, left atrium, left ventricle. So right atrium receives blood from the superior and the inferior vena cava, and the left atrium is on the opposite side here. So here we can see the openings where we have the 2 semilunar valves. Now the semilunar valves are the valves which control the flow of blood out of the ventricles. So here we can see the right semilunar valve, which only opens when the right ventricle contracts, and this prevents the flow of blood backwards when the right atrium or the right ventricle relaxes. Similarly, on the left-hand side of the heart, when the left ventricle contracts, you also have the left semilunar valve here, which opens, and when it relaxes, this closes to prevent blood from flowing back from the aorta, which is this very large diameter vessel here, back into the left ventricle. So here we have the right atrium, here we have the inferior vena cava. This is the large blood vessel which carries blood from the lower part of the body all the way back into the right atrium here. So between the atria, we have another set of valves on the left and on the right and the left-hand sides here. So this is the tricuspid valve, which controls the flow of blood so that it only flows when the right atrium contracts, and when that happens, the tricuspid valve opens to allow blood to flow from the right atrium into the right ventricle. At that point, the right atrium relaxes, the right ventricle contracts, and then this valve closes to prevent the blood from flowing back into the right ventricle, the right atrium. Similarly, when the right ventricle contracts, this valve will close while this valve opens. So there's a similar situation on the left-hand side of the heart as well. So here we can see the aorta, because the aorta actually curves downwards. So it starts at the it comes out of the heart and it curves downwards and then descends the back of through the chest cavity and then through the diaphragm down the abdomen. That is the descending aorta, and as it does so, the aorta sends off many branches. So here we have the branches which lead to the head and the neck and the arms. As it descends here, it sends off many branches to the various organs. So we saw some of those depicted on that simplified diagram of the main arteries of the body. So, for example, as it descends the abdomen, it will send off left and right branches to the kidneys, the left and right renal arteries. It will send off a branch to the liver, the hepatic artery. It will send off branches to the intestines, the mesenteric artery, and so on. Okay, so here we have the arch of the aorta. So here we have the pulmonary artery again to the left lung. So the heart is covered in a fat layer and also a membrane called the pericardium. Here we can see the two pulmonary veins which return oxygenated blood from the left and the right lungs to the left-hand side of the heart, the left atrium here. Here we see the bicuspid valve, and this sheet of muscular tissue is called the septum, the interventricular septum, or which separates the two sides of the heart. Here we can see the valve tendons, the chordae tendineae, which are responsible for holding the valves in the correct position. And here we can see the papillary muscles, which contract and relax to, again, control the opening and closing of those valves and hold those valves, valve flaps in place as the pressure rises. Now you notice here that the left ventricular wall is much thicker than the right ventricular wall. This is because the left ventricle has to generate much more hydrostatic pressure. Its function is to pump and squeeze the blood in the left ventricle so hard that it can move through the aorta and also move through all the branches that the aorta gives off to all of the different parts of the body. To do that, it needs to generate a lot more contractile force than the right ventricle, which only needs to deliver the blood to the lungs. And after all, the lungs are right next to the heart, so the right ventricle doesn't have to generate as much hydrostatic pressure. Consequently, the wall of the right ventricle is made up of a much thinner cardiac muscle compared to the wall of the left ventricle. Now, cardiac muscle is not the same as skeletal muscle. It is very different in the way that the cells appear under a microscope. They're branched rather than linear like skeletal muscles. And again, if you think about it, your heart has to constantly pump all the way through your lifetime and never get tired, whereas skeletal muscle could only maintain contractile force for a short period of time before lactic acid will build up and cause that muscle to cramp and stop contracting. So we see that the right side pumps deoxygenated blood to the lungs, and the left side pumps oxygenated blood to the body. So we actually have a double circulation, as we'll see. So the circulatory system is made up of two loops. We have the pulmonary loop, where the right ventricle pumps deoxygenated blood to the lungs, then the lungs return the oxygenated blood back to the left atrium of the heart. Then the left ventricle pumps blood through the systemic loop, which delivers blood all the way around the body, and then the blood is returned deoxygenated via the inferior and superior vena cava back to the heart. So you have two loops. So every time the heart beats, it goes through to simplify things a little bit, a three-part cycle. So the first stage, the top chambers, the atria, relax and fill up with blood from the veins. In the second stage, the atria contract and the blood is forced into the relaxing lower chambers, the ventricles. In the third stage, the ventricles contract and the blood is forced out of the heart into the arteries. So these are the contraction of the atria versus the contraction of the ventricles. So this would be sometimes referred to as atrial diastole. Diastole means relaxation, whereas atrial systole is the term used to describe atrial contraction. Similarly, we have ventricular systole, which describes ventricular contraction. Ventricular diastole would be used to describe ventricular relaxation. So blood flows around the body in a double circulatory system, as I said to you before. So this is what we referred to as the two loops, the pulmonary loop or pulmonary circuit and the systemic circuit. So here we can see that illustrated on this simplified diagram here. So obviously there are many, many more branches of the aorta to the body and many, many more branches of veins which join up to form superior and inferior vena cava, which return blood to the heart. So this is a highly simplified view of the systemic circuit of this of the circulatory system. Similarly, there are many, many arteries that deliver blood which branch off the aorta to the head and the neck. This is again very, very highly simplified. Here we can see the pulmonary loop. So we have the deoxygenated blood from the right hand ventricle pumped out of the heart via the left and right pulmonary veins, or pulmonary arteries rather, sorry. Then the oxygenated blood returns from the lungs via the pulmonary veins to the left atrium here. So this is the systemic loop. This is the pulmonary loop. So blood leaves the right hand side of the heart via the pulmonary artery, goes to the lungs where it's oxygenated. Then it travels back to the left hand side of the heart via the pulmonary vein. The left hand side of the heart then pumps the oxygenated blood around the rest of the body via the aorta. And then once the oxygen has been used up in those tissues of the body, the blood becomes deoxygenated and is returned to the right hand side of the heart via the vena cava. So this is an even more simplified view of those two loops, the systemic loop and the pulmonary loop. So this is why we describe the human circulatory system as being a double circulatory system. Okay, so you need to be able to measure the performance of the heart in sports and exercise, and this is measured in three ways. There is simply the measuring the heart rate, which is the number of the times the heart beats per minute. There is the stroke volume, which is the output of the heart during one heartbeat, but these are combined into a measure that we call the cardiac output, as we'll see. So it's about 64 beats per minute, which is a typical heart rate for a reasonably fit person at rest. So cardiac output is a combined measure of the heart rate and the stroke volume. It's the total output of blood from the heart in 1 minute. So it's calculated like this. Cardiac output is calculated by multiplying the stroke volume by the heart rate. So for example, at rest, we have a stroke volume of 75 milliliters, which is the same as saying 75 centimeters cubed. The heart rate is 65 beats per minute, and so the cardiac output for this individual would be 75 times 65, which is 4,875 milliliters, or 4.875 decimeters cubed, or liters. So while exercising, this same individual has a higher stroke volume. The heart is pumping out more blood with every beat, as you might expect during exercise. The heart rate rises also to 200 beats per minute, and so the cardiac output is therefore much higher. It's 150 times 200, which gives us 30,000 millimeters, or 30 decimeters cubed, being delivered per minute. So let's skip past this. Okay, so a number of things could go wrong with the circulatory system. Now, by far the most common form of cardiovascular disease arises as a result of a process called atherosclerosis. Atherosclerosis is the buildup of fatty deposits in the walls of arteries due to, for example, too much cholesterol over a long period of time. So high levels of cholesterol can narrow the arteries and bring about increased risks of myocardial infarctions or heart attacks and other forms of heart disease. So here we have a diagram showing a healthy blood vessel with a clean, shiny looking lining, and this in contrast would be what it would look like with cholesterol deposits building up in a unhealthy individual. And so here we can see that not only is the diameter of the artery through which the blood flows much narrower, this also causes other conditions like high blood pressure, which itself can cause a lot of damage to various organs of the body, including the heart. But also we can see here that the lining has become rough, and this causes a turbulent flow of blood through the vessels. The heart also has to work much harder to force blood through these much narrower arteries, and if the coronary arteries, which are the arteries which branch off the aorta and provide oxygenated blood to the heart muscle itself, if those become blocked up by these fatty deposits, then that can lead to the death of regions of the heart muscle itself due to lack of oxygen, and this is what leads to heart attacks. So one of the ways that this can be treated is to insert a device called a stent into the artery. This device is essentially just a little balloon which can then be inflated from the outside to open up the artery, to widen the artery. So the narrowing of arteries contributes to the onset of cardiovascular disease by reducing the blood supply to the heart tissue itself. The tissue becomes weak and a heart attack may result from that. So the cholesterol deposits or plaques can also cause problems by breaking away and floating through the bloodstream, and this can also cause strokes and heart attacks when those blockages arise in the coronary vessels and in the fine arterioles, which supply various parts of the brain as well. So this process of atherosclerosis can lead to all kinds of different very serious medical conditions. So here's how a stent works. You essentially have this very long wire at the end of which you have a inflatable balloon with coated with drugs. So this is inserted usually into an artery in the neck and then fed through the various arteries on the way to the blocked artery of the heart, and the operator can see what they're doing because the person can have external imaging applied, like an X-ray or some other form of imaging, to show where the stent is as they feed it through that artery. They can position that stent into the blocked vessel, inflate the balloon, and this then widens the blood vessel. So another treatment, a rather drastic treatment for extreme cases of heart disease, is to replace the human heart entirely. So the first human to receive a heart transplant received it in 1967. So the donated heart came from a recently deceased donor. So today there is a 70% survival rate for at least 5 years or more after the operation. Now, part of the reason why it's not 100% is that when you receive a donated heart, even if it's correctly tissue matched to your own tissue antigens, your immune system is extremely good at spotting differences in your antigens versus the antigens of other hearts. And so eventual tissue rejection is one of the causes of death from this, but also simply failure of that heart because it is a rather invasive and drastic surgery. So, the heart only really has one function, to pump blood around the body, so it is quite logical that its function could be replaced by a mechanical pump that could be developed to replace the natural heart. But these kinds of mechanical pumps would require a continuous source of power and would also have to respond to the varying demands of that human being as they, for example, are exercising or doing more physical activity. So, Dr. Robert Jarvik implanted the artificial Jarvik 7 heart into a dentist, Barney Clark, in 1982, and Mr. Clark only lived for another 112 days after the operation. So in 2001, after about 30 years of research and development by Biomed, which is the world's first complete self-contained heart replacement called the BioCore, was implanted into a 58-year-old male patient. Now, this is intended for patients with end-stage heart failure who have no other options for treatment. So the second version of this device is being developed for production and did actually go into production later on. Okay, so another thing that can go wrong with hearts generally is that the valves can undergo dysfunctions. You can have problems with the way those semilunar valves and also the atrioventricular valves, the tricuspid valve on the right-hand side of the heart and the bicuspid valve on the left-hand side of the heart. These are moving structures, and you can either be born with heart valve defects, or heart valve defects can develop later in life. So there are artificial heart valves which can be used if the heart valve fails due to disease or inherited disorders. And these can be life-threatening. Modern medicine can replace any one of those 4 heart valves. Now, the replacement valves can either be mechanical or they can be made up of actual tissue. And mechanical valves can last a lot longer than a human lifespan and don't need replacing. So these are actually quite successful prostheses. But mechanical heart valves all require lifelong treatment with anticoagulant drugs, which are sometimes called blood thinners, to prevent the formation of blood clots, which can be potentially life-threatening. So tissue valves, which are made up of pig heart valves, can last up to 15 years before they require replacement. Now, this has to be done by open heart surgery, which again carries its own set of risks. Now, tissue heart valves, these in contrast to the mechanical ones, don't require regular treatments with anticoagulants to maintain the life of the patient. Now, the reason for this is because of the improved blood flow. Mechanical valves cause turbulent blood flow, and again, as we saw in that example of atherosclerosis, when you have turbulent blood flow inside the circulatory system, the risk one of the risks is that this can cause embolisms, which are blood clots inside the circulatory system, which can then float through the circulatory system, eventually lodging in smaller arteries and blocking the supply of blood to regions of the brain or regions of the heart, which cause strokes and heart attacks. And so these pig heart valves have a lower risk of that happening. So we also have heart-lung machines. Now, heart-lung machines are this is a device which is used during operations on the heart itself. Now, sometimes medical procedures need to be done. Surgery needs to be done on the heart, for example, which requires the heart to be stopped so that it's not pumping blood. So these heart-lung machines actually replace the function of the heart's pumping action, and also replace the lungs by adding oxygen to the blood artificially. So the heart-lung machine takes blood from the upper right chamber of the heart, the right atrium, and after it's been treated with oxygen, it's returned directly by the machine to the aorta. So it's kind of bypassing the heart, which allows the heart to be stopped artificially so that surgery or other procedures can be done on the heart. So from the aorta, the oxygenated blood then moves on to the rest of the body as usual. So here we can see a heart-lung machine in operation during a heart operation. Now, heart operations are extremely delicate procedures and can last for several hours, but the longer the patient is connected up to the machine, the more likely the patient is to suffer side effects. So heart-lung machines are commonly used in operations like these in developed countries, but again, they do carry their own set of risks as well. So all of the cells of the body require energy in order to work properly. Muscle cells would not be able to contract without a supply of oxygen and glucose, for example, and so the circulatory system acts as the body's transport system. It carries blood, which itself carries oxygen and glucose, which are required for respiration, but also carries waste products away from respiring tissues, including carbon dioxide, for example, and also urea. So it's essentially a delivery service and a waste disposal system. So adult humans have about 5.5 litres of blood on average, and so blood itself is a tissue. It's made up of various kinds of cells, which are specialised to perform a particular function. So if you take blood and leave it to and take blood from a patient and enter it into a centrifuge, we find that it can separate into four parts. We have the blood plasma. So blood plasma is the liquid components of the blood once you remove cells from the blood, and also so once you remove cells from the blood, you have plasma. Once you remove the large plasma proteins from the plasma, you have another type of fluid called serum. What you also have, once you allow the centrifuge to separate out the components of the blood, you have platelets. Now, these are cell fragments which are responsible for blood clotting. You also have white blood cells, which are responsible for immune responses. And finally, you have red blood cells. Now, the red blood cells make up about 45% of the total volume of the blood. So here we can see a photomicrograph at 1,000 times magnification showing a blood smear. And so this shows you the appearance of blood under a microscope at 1,000 times magnification. So we see here red blood cells, we see also white blood cells here, and here. We see the blood plasma, which is the liquid component between the blood cells. And we also see some fragments of blood cells called platelets as well. So plasma is the liquid part of the blood. When you remove the blood cells from blood, you end up with a straw-coloured suspension and solution called plasma. So its main role is the transport of carbon dioxide, the transport of dissolved substances like glucose and amino acids, and urea, which is a metabolic waste. The other role is actually heat, kind of thermal transport. The cells of the body are constantly respiring, and that process of releasing energy from food is not particularly efficient. In other words, a lot of the energy released from food substances and also released from ATP to power cellular processes ends up as thermal energy, as heat. Now that heat has to be removed, otherwise the temperature would rise to the point where those cells would die due to the denaturation of their proteins. And so the blood, remember, every cell of your body is no more than 1 or 2 cell widths away from a capillary, and so the flow of blood through the capillaries takes heat energy away from cells so that those cells essentially don't bake in their own waste heat. So white blood cells are nucleated cells, and their role is basically protection. They can engage in phagocytosis. They can destroy invading microbes. They can produce antibodies, which are chemicals, proteins, which are responsible for attaching to specific antigens on microbes to destroy them. We also have platelets, which are responsible for blood clotting, or take part in the process of blood clotting. These are cell fragments, and their main role, as I say, is to help clog a meshwork of fibers which develops when you cut the surface of your skin or break capillaries, and this prevents bleeding. So the red blood cells have no nucleus. They are shaped as biconcave discs, and they're also very flexible, so they can fit through those bendy capillaries and flow single file through them. Their main role is to collect oxygen at the lungs and then transport that oxygen to the cells of the body for respiration. Now, in order to do this, they contain a substance called hemoglobin. Now, hemoglobin bonds very effectively at high oxygen concentrations, like in the lungs, to oxygen and becomes oxyhemoglobin. Then, when the hemoglobin in the red blood cells is transported to the tissues where the concentration of oxygen is very low, the oxygen then unbinds from the hemoglobin and diffuses out of the capillaries into the surrounding tissues. So here is a false color image of a scanning electron micrograph. Here we can see the biconcave disc shape of the red blood cells. Now this shape maximizes their surface area to volume ratio for effective diffusion of oxygen outwards at the tissues and into the red blood cells at the lungs. So the next topic that you'd like, that you all, not all, but most of you voted for in the resources room or the resources page for me to go over is the structure and function of the digestive system. So the purpose of digestion, digestion is just one of several stages that are responsible for the obtaining of nutrients from the food that we eat directly to the cells where those nutrients are required. So this diagram kind of summarizes the main stages in that. We have ingestion, the taking in of food into the mouth. We have mastication, which is the cutting and grinding of the food into smaller pieces to provide a higher surface area over which chemical digestion can take place. We then have digestion. Now, digestion includes physical digestion like mastication, but chemical digestion also occurs, whereby enzymes, digestive enzymes, break down the biological macromolecules like proteins, nucleic acids, complex carbohydrates like starch and lipids into their smaller building block molecules, their smaller monomers like amino acids and so on. So, digestion is the process generally by which food is broken down into soluble materials, soluble small molecules by physical digestion and also by digestive enzymes. You then have the process of absorption, which happens predominantly in the small intestine, whereby the small food molecules into which the larger ones have been digested are absorbed directly into the bloodstream. We then have assimilation. Now, soluble digested food in the bloodstream is carried again to capillary beds, where it diffuses out of those capillaries into the tissue fluid and then dissolves and is taken up by the cells of those tissues. So this is assimilation here. And finally, we have the removal of undigested food, which is egestion. In the case of human beings, this involves the removal of food, of undigested food from the rectum via the anus. So this is called defecation. Okay, so I see that we are coming up towards the end of the first 55-minute section of this webinar, so I'm going to take a brief break here for 5 minutes. Again, at the end of this 5-minute break, you should be able to see a poll pop up on your screen, those of you in attendance, and you'll be able to vote as to whether you want me to continue with the topic teaching or whether you'd like me to go through the exam style questions here. Obviously, either way, please do cast your vote, and whichever of those receives the most votes is what we will do after this 5-minute break. So take a breather for about 5 minutes, and I will see you shortly to continue the webinar. Okay, everyone, we're back after the 5-minute break, and I see that so far we've got quite a few very good questions being asked in the Q&A boxes for the 5 Zoom webinar rooms. Keep those questions coming, guys. As you might have noticed, there haven't been any pop-up questions so far in this webinar, just because there are so many things to get through. I'm kind of reserving those for this section of the webinar here, should you guys wish to continue. So let me just take a look at the results of that poll, and we'll see what we're going to be doing for this next hour or so. Okay, so it seems like most of you, approximately 67% of you, or thereabouts, have voted for me to continue with the topic teaching, please do be aware that I will be uploading the full mark scheme, as always, for the exam style questions that you received in your webinar pack to the private resources page within about 48 hours of the end of this event. So if you have any questions about any of those, please do type those either in the chat on that page or send me a private message through that page, and I'll respond usually within 24 to 48 hours. And I will be continuing to respond to questions and providing revision support for you, the attendees, all the way up to the end of your exam period in June later on in June. So let's continue with what we were talking about with ingestion, digestion, absorption and assimilation. So for example, absorbed sugars can be used immediately for respiration, but many molecules will be assimilated, built up into macromolecules such as new proteins, glycogen or fats. So digestion, we have large food molecules being broken down by digestive enzymes. We have absorption and assimilation. And we also have a network of blood vessels around the lumen, the lining of the small intestine, for more efficient absorption of the digested products. And finally, the undigested food substances are removed during defecation. And so the removal of undigested food is referred to as egestion. So the human digestive system is made up of a series of organs and accessory structures. So we have the buccal cavity or the mouth, we have the esophagus, we also have the liver, the large intestine, the rectum, which stores the undigested food so that it can be egested later on. We also have accessory structures such as the salivary glands, we have the stomach, we have the pancreas, which is a leaf-shaped organ which is found in the loop of the first section of the small intestine called the duodenum. We have the major portion of the small intestine, the duodenum and the ileum, as well as another section called the jejunum. And we have the anus, which is the opening through which the undigested food is egested. Now, the position of the liver is shown in this surface marking here, and here we can more clearly see the esophagus going through the emptying into the upper portion of the stomach. The stomach then empties via the pyloric sphincter into this first section of the small intestine. This is called the duodenum. Now the rest of the small intestine is highly simplified here, but this gives you a sense of the structures and their relative positions. So here we can see a cutaway section of the large intestine to show the duodenum behind it. So we have the position of the liver. The liver also has the gallbladder attached to it. This is where the bile is stored. Now the liver manufactures this liquid called bile, which contains bile salts, and then stores it in the gallbladder before releasing it via the bile duct, which joins onto the pancreatic duct and releases, and those two ducts empty into the duodenum of the small intestine. Excuse me. So now the purpose of bile is to neutralize the acidic contents of the stomach as they empty into the first portion of the small intestine, the duodenum. Now the reason for this is that the acidity in the stomach provides the correct pH for the stomach enzymes, the pepsin, to work and break down proteins, but the enzymes which work in the small intestine require an alkaline pH, and so the acid from the stomach needs to be neutralized in order to provide the correct pH environment for the pancreatic enzymes and the enzymes secreted by the walls of the small intestine to work properly. And so this is part of the function of bile. The other function of bile is to emulsify lipids. So you have large lipid droplets in the contents of the stomach which empty into the small intestine. Those are broken up into much smaller droplets, and this maximizes the surface area of the lipids over which the lipase enzymes can break them down. So here we see this leaf-shaped organ, the pancreas, indicated by the dotted line here. This is the duodenum, and we have a simplified view of the ileum here. Now the small intestine joins up to the large intestine, So we have here the ascending colon, the transverse colon, and we have the descending and sigmoid colon here. But there is a structure, a vestigial structure called the appendix, and it's the appendix which becomes inflamed during appendicitis. Now, if we zoom in on this region here, we can see a little bit more detail as to the structures in this region. So here we can see a cutaway section of the stomach, which shows us the layers of muscle in the stomach wall. This is the gallbladder, and this is the bile duct here. So here we have the gallbladder, which is positioned next to the liver. The bile duct, here we can see the pyloric or lower sphincter, which controls the movement of food from the stomach into the small intestine. Now the food sits in the stomach for 3 or 4 hours, during which protein digestion starts to take place, after which the pyloric sphincter relaxes and allows the chyme to move into the small intestine here. So here we can see the lower end of the esophagus, and at this end of the esophagus, there is also another sphincter, which is called the cardiac sphincter, or the upper sphincter of the stomach. This controls the movement of the bolus of food from the esophagus into the stomach here. So here's the stomach, and here we can see the longitudinal and circular layers of muscle. Now the reason why the stomach has a very thick muscular wall is because the stomach wall itself contracts rhythmically to churn up the food in the stomach and mix it with the digestive enzymes and the acid which is secreted by the stomach walls. So here we can see the pancreas, and we can see the pancreatic duct as well here. So we see here where the bile duct joins up with the pancreatic duct, and then both of them open into the small intestine at the duodenum. So the bile and the pancreatic juices containing various pancreatic digestive enzymes and other substances, these mix together and then empty into the lumen of the small intestine here. So the walls of the alimentary canal, or the gastrointestinal tract, which is a kind of general term for the whole digestive system, typically do contain layers of muscle like you see here. Now the small intestine, for example, also contains the wall of the small intestine, which is simplified here, actually is made up of layers of smooth and longitudinal muscle. Now the purpose of this is for a process called peristalsis. Now peristalsis is a type of involuntary contraction in the intestines and the colon, but also in the esophagus. Now through this type of contraction, the longitudinal fibers of muscle and the circular fibers of muscle in those walls contract behind the ball of food, which is called the bolus, and push the bolus along those parts of the small intestine and also the large intestine and also the esophagus. So in this way, the food passes through each area of the digestive system, exposing the food to a variety of digestive enzymes and conditions as well. So I've mentioned digestive enzymes quite a lot, and this is one of those other topics that many of you voted for me to cover. So let's take a look at that. Now, before I do so, let me just quickly ask a question. So you should see this question pop up on your screen. If you don't, please do type it into the webinar chat so I can see your responses. So what do we mean by an enzyme? Okay, so most of you answered the question correctly. Yes, enzymes are metabolic catalysts. Now, some of you selected the response where enzymes are chemicals which digest food and break food down. Now, that is true, but not true of all enzymes. Enzymes generally are chemicals which catalyze biochemical reactions and speed them up. So, in terms of digestive enzymes, we're going to be looking at enzymes more generally shortly. The sources of digestive enzymes and the locations where they're secreted in the digestive system are the mouth, where you find salivary glands. These manufacture and secrete amylase, which is an enzyme that breaks down carbohydrates. So it's called a carbohydrase. Now the term carbohydrase applies to many different enzymes, as we'll see, but this is amylase. This is the carbohydrate secreted in the mouth, and this breaks down starch, which is a polysaccharide, into a disaccharide called maltose. So saliva production is stimulated through nervous activity. So the nervous system detects the presence of food or the thought of food, and this causes salivation, the secretion of saliva by the salivary glands. So salivation can be triggered by the presence of food, the sight or smell of food, or even the thought of food. So here we can see a diagram showing the structure of starch. Each one of these little circles represents a glucose molecule which have been linked together in these branched chains. Now, in order to break down this molecule and release smaller glucose molecules, first of all, we break it down into larger segments called dextrins. Eventually, those are broken down by amylase into disaccharides, maltose. So starch digestion begins in the mouth, where 3 pairs of salivary glands in various portions of the, or rather located in various positions around the mouth, secrete saliva into the mouth, and the saliva contains amylase, and this catalyzes the breakdown of starch into a disaccharide called maltose by the hydrolysis of glycosidic bonds. Now in the stomach, gastric glands secrete proteases such as pepsin into the lumen of the stomach. Now, the gastric glands also secrete hydrochloric acid into the stomach. Now, one of the functions of hydrochloric acid is to provide the correct low pH for the functioning of the activities of various protease enzymes. So trypsin, pepsin, and so on. So pancreas, the pancreas produces a number of secretions, but it's also actually involved in the immune system as well. The pancreas contains the islets of Langerhans, where you find the alpha and beta cells. Now these are responsible for secreting hormones, hormone molecules which control the concentration of sugar molecules in your bloodstream, but that's something we're going to cover in a later webinar in this series. So the pancreatic juice, which is secreted by the pancreas into the duodenum of the small intestine, contains a number of different enzymes. It contains carbohydrases, it contains proteases, it contains lipases. So here is the pancreas, and it secretes its pancreatic juice into the duodenum, the first part of the small intestine. Now the pancreatic juice also contains hydrogen carbonate ions. Now these also neutralize the acid from the stomach along with the bile. So the walls of the small intestine itself also contain glands which secrete a number of other digestive enzymes. So proteases, lipases, carbohydrases are also secreted by the walls of the duodenum. So the walls of the small intestine do contain circular and smooth longitudinal muscles. So here we can see the circular muscle layers, the longitudinal muscle layers, but the inner lining of the small intestine is highly folded into finger-like projections called villi. Now these provide a very large surface area over which the secretion of digestive enzymes, but also the absorption of small molecules produced by the digestion of these biological macromolecules can also take place. So if we magnify this, we see villi. If we further magnify these villi, we would also see on their surfaces microvilli. So this is the small intestine and a single villus. Villus is the singular, villi is the plural. So we see here goblet cells in the surface of the epithelial lining. These secrete mucus, and the mucus lubricates the surface, the lining of the small intestine to allow the food to pass through the gut more efficiently. Here we can see this highly branched network of capillaries responsible for absorbing the digested food products. We also have lymphatic vessels in the middle of these villi. So these lacteals join up to the lymphatic system. So the lacteals absorb undigested oil droplets, but also the digested products of lipid digestion, fatty acids and glycerol, into the lymphatic system, which then transports them around. Here we have the blood vessels, which eventually join up into venules, which eventually join up into the hepatic portal vein, which transport the digested food products which have been absorbed to the liver, and eventually those return to the heart to be pumped around the body again. Here we see the small intestines glands, the glands in the walls of the small intestines. Now these are the glands responsible for producing the digestive enzymes, the proteases, the lipases, and the carbohydrases, which the small intestine produces. So we mentioned the gallbladder, but pancreatic juice also contains hydrogen carbonate ions, so some of the same functions are served by the pancreatic juice as well as the bile, which is stored in the gallbladder. But the bile salts, the chemicals in the bile responsible for the emulsification of fats, as well as the alkaline pH, these are produced. The bile itself is produced by the liver but stored in the gallbladder. So what you're not seeing here is the liver, which is this organ on the right hand side here. This is what produces bile. It's stored in the gallbladder. So we see the gallbladder, the bile duct, and the bile duct again joins with the pancreatic duct and opens into the small intestine here. So carbohydrates, these are polymers of simple sugars such as glucose and fructose. Now these bonds between the monomers, the monosaccharides, can be broken by enzymes by hydrolysis to release simple sugars. So starch is a major source of carbohydrate in the human diet, and it's digested eventually into small glucose molecules which can be absorbed. So these are typical sources of starch, and starch itself is a mixture of two polysaccharides, polymers of glucose, amylopectin and amylose, which is a spiral molecule. So starch molecules, because they're very large, they are insoluble, and so they cannot be transported directly from the lumen of the small intestine into the blood. They first need to be broken down into smaller units, glucose molecules, which are soluble and can be absorbed into the bloodstream. So this diagram indicates that we start with large starch molecules, which are small glucose molecules chemically bonded together. These are digested first by amylase and eventually by other enzymes to form individual glucose molecules, which can then be absorbed through the gut wall by the epithelial cells lining the gut into the capillaries within those villi. So proteins are made up of monomers called amino acids, which are joined to each other by peptide bonds, but peptide bonds can also be hydrolyzed by digestive protease enzymes to form the individual amino acids that are small enough and soluble enough, again, to be absorbed via the epithelial lining of the gut directly into the bloodstream, where they can then be transported to where they are needed. So protease enzymes, like all enzymes, are specific and will only digest protein molecules. So many enzymes are so specific that they'll only break down certain parts of protein molecules. So you have endopeptidases and exopeptidases. So endopeptidases are protease enzymes that will break bonds in the middle of a polypeptide chain, in the middle of a protein, which is made up of chains of amino acids. Now these different amino acids are shown rather simplified here like this, because they are different shapes, but they are bonded by the same types of chemical bonds. So these enzymes, protease enzymes, can either break bonds in the middle of the molecule, or they can break bonds at the ends of the molecule. So enzyme B will only hydrolyze the peptide bonds between specific pairs of amino acids. So the specificity of enzymes' active sites, which we'll talk about shortly, ensure that only one particular substrate molecule can lock into it and react. So several different protease enzymes are required to fully digest any particular protein into its component amino acids. And so, in fact, simply saying the term protease, you're not referring to one particular enzyme, you're referring to a whole category of different enzymes. So proteins, once again, are polymers. They are large molecules made up of repeating monomers, repeating individual smaller units, which are chemically bonded together. In the case of proteins, these are amino acids. The monomers are amino acids, and these are held together by bonds called peptide bonds. Now, a peptide bond is shown in this diagram here. So you have the first amino acid monomer, you have the second amino acid monomer, this section of the bond of the molecule here, this is the peptide bond. Now, when you hydrolyze this bond, hydrolysis means the breaking of a bond by adding, excuse me, adding a water molecule across the bond here. So, when you hydrolyze this bond, you get the separate amino acids. So the H2O, the 2 hydrogens and the 1 oxygen from this water molecule have been added across this bond to break the bond. Now, similarly, amino acids can be assimilated by your cells into proteins by joining them together in the opposite direction. So the opposite process is called a condensation reaction. Okay, so let's take a look at the way that fats and oils, or lipids, are now digested. So, fats and oils are digested into much smaller molecules called fatty acids and glycerol. Now, these can easily be assimilated and combined again into lipids by the cells of the body. So, this is the structure of a fat molecule. This is the glycerol part, and you have 3 fatty acid chains. Now, again, this is the breaking of these bonds is done by hydrolysis. We have 3 molecules of water which are added across here to form these hydroxyl groups. So this is glycerol, propane 1,2,3-triol, and you have 3 fatty acid chains. So we've reformed the carboxylic acid functional group here by adding water across these bonds. So you add H and OH here. So H goes here, OH goes here. That's 1 water molecule. The same thing happens here and here and here to form 3 molecules of fatty acids and your single glycerol. So this is a triglyceride, a fat, and they can be broken by hydrolysis by lipase enzymes to form 3 fatty acid molecules and 1 glycerol molecule. Okay, so to summarize, this table shows you the locations of carbohydrases and their functions, the locations of proteases and their functions, and the locations of lipases and their functions as well. So let's take a quick look at enzymes in general and the ways that enzymes work, because this is one of those topics that many of you voted for me to go. So, enzymes are metabolic catalysts. Many of the chemical reactions that occur in living organisms would not occur anywhere near fast enough to sustain the living processes of those organisms, or they wouldn't even happen at all if you simply mix those reactants together in a test tube in a laboratory. Biological catalysts called enzymes are chemicals which speed up these biochemical reactions. Now, the word catalyst on its own just means a substance which speeds up a chemical reaction by enabling that reaction to proceed at a lower activation energy or more easily. So catalysts are unaffected by the reaction because the catalysts themselves are not reactants. They don't participate directly overall in the reaction, and they can be recovered and take part in further reactions after the products of the reaction have unbonded from them. So the word catalyst generally has this meaning in chemistry, but in biochemistry, the types of catalysts that you find in biological systems are enzymes. So enzymes are catalysts because catalysts in general work by lowering the activation energy, and this is what enzymes do. They speed up biological reactions by lowering the minimum energy necessary for the reactants to combine chemically with each other to form the products. So enzymes are sometimes referred to as biological catalysts, and these are catalysts produced by living things. So the molecule on which the enzyme acts is called the substrate, and the substance produced as a result of an enzyme catalyzed reaction is called the product. So enzymes are specialized proteins. So enzymes themselves are protein molecules with complex 3-dimensional shapes, and like all protein molecules, they are made from the 22 naturally occurring amino acids. So again, proteins like carbohydrates and nucleic acids like DNA and RNA are biological polymers. They are large molecules made by linking together chemically smaller building block molecules called monomers. In the case of enzymes, they are proteins. So like all proteins, they are composed of complex 3-dimensional shapes, which ultimately arise as a result of a linear sequence of amino acids being chained together and then folding up in complex ways. So this is the enzyme fluorinase, and this is a computer-generated diagram showing the structure of that enzyme. So what you're seeing here is lines which indicate chains of amino acids, and these coils indicate when that linear chain of amino acid coils up, and then these sheets indicate when the amino acid chain folds up like this into a sheet. And so the overall three-dimensional shape of the enzyme is what gives it its function. Now, remember, enzymes are not living things. This is a mistake that I see a lot of students making. Enzymes themselves are not living. They are components. They are organic molecules made by living things that perform specific functions. So each of the thousands of different chemical reactions that occur in living things has one or more enzymes which speed them up, which catalyze them. So here we can see the three-dimensional computer-generated models of different enzyme molecules which all have different functions. So acetaldehyde dehydrogenase dehydrogenates, removes hydrogen from a chemical called acetaldehyde. Alcohol dehydrogenase removes hydrogen and breaks down alcohols. Catalase breaks down hydrogen peroxide. Fructose biphosphatase breaks down fructose bisphosphate. Pepsin breaks down proteins. Maltase breaks down carbohydrates. Amylase breaks down carbohydrates as well. So these, you can see, have radically different structures because they break down. They have different substrates that they attach to. So again, a catalyst generally causes reactions to happen more quickly, and it does this by providing an alternative way in which the reaction can take place, which has a lower activation energy. So if we have an exothermic reaction where the reactants are at a higher energy level than the products, without the catalyst, the activation energy barrier, the amount of energy which you need to provide to the reactants to get them to the point where they will react and form the products, is higher without the catalyst. With the catalyst present, the activation energy barrier is lower, and so the products can be formed more easily with less energy input. So some enzymes build up more complex molecules from simpler ones. So this is another misconception that I see from students all the time. Enzymes are not just chemicals which break down other substances. You might think that from listening to me talk about digestive enzymes, because digestive enzymes are involved in digestion, the breaking down of substances. But enzymes can also build substances up. The amino acids, the glucose molecules, the fatty acids and glycerol that you absorb in your digestive system and gets carried around in your blood to the tissues that need it, they need to be reassembled into the proteins, into the starch, into glycogen, into the lipids that your cells need. And so there must be other enzymes that take those smaller molecules and build them back up into larger molecules. These are referred to as anabolic enzymes. So some enzymes are anabolic, some enzymes like digestive enzymes are catabolic. They break down substrates into smaller molecules. So this is the general way of categorizing enzymes into their functions. They are either enzymes that build up larger molecules from smaller ones in the case of anabolic enzymes, or they are catabolic enzymes. They take a substrate molecule which is large and break it down into smaller building blocks. So salivary amylase that we mentioned before is found in saliva. It's a catabolic enzyme. It converts a large molecule starch into a smaller disaccharide molecule called maltose. So this is an example of a catabolic enzyme. So enzymes work first by bonding to a specific substrate, and so they have special sites on their surfaces called active sites. So this is where the reacting substrate molecules fit into them. Now, we mentioned before that enzymes are highly specific. A particular enzyme like pepsin, for example, will only work on a particular substrate, a protein in the case of pepsin. If you mix pepsin with a complex carbohydrate like starch, it will not break it down. So enzymes are highly specific, and the reason for this is because each enzyme, as we saw in the previous molecular models, have highly specific three-dimensional shapes which have highly specifically shaped active sites which will only fit particular substrate molecules. So an amylase enzyme will accept starch molecules but not the molecules of other carbohydrates or proteins or lipids. So active sites are actually complex structures themselves, and one enzyme molecule will actually often have several active sites, and of course this will help speed up the reaction even further. So the active sites in the molecules of fluorinase enzyme, which is found in some soil microbes, are shown in this diagram. So how many active sites do you think you can identify in this enzyme? So I'll leave that up there for a second or two, and I would be interested in hearing your thoughts on that in the live chat on the resources page. So one of the things that many of you have been asking about via private messages is the lock and key hypothesis and the induced fit model some of you have been asking about, which is actually quite encouraging. So substrates and enzymes react together specifically. So the lock and key hypothesis, or the lock and key model, is a scientific model which attempts to explain this observation of the specificity of enzymes. So substrate molecules and enzyme molecules will react together in a specific manner. And the reason for this is because of the complementarity, the complementary shapes of the enzyme molecule and the substrate molecules. So in other words, the enzyme will have a active site, one or more active sites, which has the exact right shape necessary to fit a particular substrate molecule. So when that substrate molecule fits into the active site, this causes the chemical reaction to occur, which then produces the products which diffuse away from the active site, leaving the enzyme ready to accept another substrate molecule. Notice that the substrate molecule has been chemically changed to produce new product molecules, but the enzyme has retained its shape and active site shape because the enzyme is a catalyst. It can go on to catalyze the reactions of other substrate molecules into products. So in this way, a particular enzyme will only bind with a particular substrate, and this is what we call the lock and key model of enzyme activity. So one of the things that you do need to understand is the effects of different environmental variables on the rate of enzyme action. So let's start with the effect of temperature on enzyme activity. So at low temperatures, the reacting molecules of enzyme and substrate have lower kinetic energies. Temperature is a measure of the average kinetic energies of the particles of a substance. So if you have a mixture of enzyme and substrate molecules at a lower temperature, they are moving less rapidly. That's what temperature means. And so they are colliding with each other and bonding with each other and then reacting to form the product at a lower rate. If we compare this to a higher temperature, the particles are moving more rapidly, and so by random collisions they will form the products more rapidly. The rate of enzyme reaction, enzyme catalyzed reaction, will be higher. So if we compare these two, we see that at higher temperatures the rate at which the products are formed from the reactants is higher than at lower temperatures. So once again, temperature is a measure of the average kinetic energies of particles, and so the higher the temperature, the higher the rate of collision and bonding between the enzyme molecules and the substrate molecules, and therefore the higher the rate of the reaction. However, eventually a temperature is reached where the rate of reaction is greatest. This is called the optimum temperature. But because enzymes are proteins, and proteins will become denatured at a temperature which is too high, as the temperature is increased beyond the optimum temperature, the special bonds that stabilize that complex three-dimensional structure of the enzyme become broken and the structure becomes disordered. This is what we call denaturation, and therefore the active site of the enzyme loses its complementary shape and therefore cannot catalyze the reactions of the substrate into the products. So at this point, the substrate molecules no longer fit the product because the enzyme molecule has changed shape, it has denatured. So denatured enzymes will not be able to catalyze a reaction, and so the products will not be formed. Now, denaturing is permanent. The random movement of the enzyme so that it disorders itself, it means that you lose that very specific shape. And so reducing the temperature down again will not undo the damage to the active sites. So as the temperature increases, molecules move more quickly and collide more often, so the reaction rate increases. Now, in general, the reaction rate will double for every 10-degree rise in temperature until you get to the optimum temperature. So at the optimum temperature, we have the highest possible rate of reaction for that enzyme with its substrate. And then if you increase the temperature beyond the optimum temperature, the rate of the enzyme catalyzed reaction rapidly decreases because the enzyme molecules are becoming denatured. So each enzyme has an optimum temperature where you get the fastest rate of reaction, but in a similar way, each enzyme will have its own optimum pH at which you get the fastest reaction rate. pH can alter the shape of an active site if it goes too high or too low. So the shape can be temporary. The shape change, again, denaturation can be triggered by conditions which are too acidic or too alkaline. And so we see here that if we increase the pH from an acidic pH to an alkaline pH, like with temperature, you do get an optimum pH level. But this is different for different enzymes, and this is why in the digestive system, different enzymes require different pHs to work at their optimum rate. So pepsin, which you find in the stomach, works at an optimum pH of 2 pH units. So this is very, very acidic. This is why one of the reasons why you have acid in the stomach. The other main reason you have acid in the stomach is to kill microorganisms which may be present in your food. Now, once the stomach contents go into the small intestine, you need an alkaline pH because you have lipase and amylase. Now, amylase has an optimum pH of about 7.2 in the mouth, and so the saliva in the mouth is very weakly alkaline for this reason. But lipase, which you find in the small intestine, requires an alkaline pH, a highly alkaline pH. So this is the function of the hydrogen carbonate ions in the pancreatic juice, and also the ions that you find in bile. It is to neutralize the acid contents of the stomach and then cause that solution to become alkaline so that enzymes like lipase can operate at their optimum rate. So inside living cells, not just in the lumen of the alimentary canal and the digestive system, you also have many, many different chemical reactions inside cells which are catalyzed by enzymes. Now, some of the most important of these are the enzyme catalyzed reactions of respiration, but there are many others. So there's protein synthesis, there's photosynthesis, intracellular digestion. So during aerobic respiration, in other words, respiration in the presence of oxygen, there are many, many different enzyme catalyzed reactions which occur. So these, for example, use sugar and oxygen and release energy. But there are many, many other internal reactions that occur inside cells which require enzymes to catalyze them. In fact, almost all biochemical reactions that occur inside or outside of living cells require one or more enzymes to catalyze them. So enzymes are extremely important for that reason. Okay, so the next thing that you all voted for me to go through is the food tests for different nutrients, different food groups. So let me pause here and ask you, the audience, another question. So you should see this question pop up on your screen, those of you in attendance again. If you don't, then please do just go ahead and type your question in the Q&A panel, rather in the chat box. Leave the questions in the Q&A panel for the ones that you'd like me to answer in depth after the webinar concludes. So here's your question. What are the different categories of carbohydrates that I've mentioned already? And give some examples of each. Okay. So, very good. A lot of you have gotten the correct answers here. So let's quickly go through how carbohydrates are classified, and then we'll go into how we can test for various different kinds of food groups, including carbohydrates. So, firstly, carbohydrates are generally classified into either sugars or polysaccharides. So saccharide refers to molecules of sugar, poly means many. So polysaccharides can further be subdivided, but also sugars can further be subdivided into subcategories. So sugars are either monosaccharides like glucose or disaccharides. So single sugar units, the simplest type of sugar molecules, are called monosaccharides. Disaccharides are made by linking together two molecules of a monosaccharide into a slightly larger molecule. So di meaning two, mono meaning one, saccharide referring to a sugar molecule, a small sugar molecule. Polysaccharides can be divided up functionally by their function into either storage molecules, which store large numbers of small sugar molecules chained together, or structural molecules, which are intended to provide structural support. Now, these are typically found in plants and fungi and bacteria. So monosaccharides are single sugar units. Now, this includes glucose and fructose. So here's a ball and stick model of a glucose molecule. Disaccharides are double sugar units of two molecules of sugar, like a glucose, joined together by a glycosidic bond. So these are things like sucrose, which is common granulated sugar that you find in the kitchen, but also maltose and lactose. So glycogen in animals is the storage molecule, the storage carbohydrate, and starch is the one found in plants. So cellulose is the structural polysaccharide found in the cell walls of plant cells. This provides structural support to the cell, and also chitin is an important structural carbohydrate found in the exoskeleton of arthropods. So how do we test for reducing sugars? Now, all of the monosaccharides and many of the disaccharides are classified as reducing sugars. So the Benedict's test is used to determine whether or not a particular sugar is a reducing sugar. Here is a photograph of blue Benedict's solution. So this is a turquoise blue solution that contains a mixture of copper ions and sodium ions and hydroxide ions. Now the copper ions exist as copper 2 plus ions in this reagent. Now if you mix this with a reducing sugar, then the copper 2 ions are reduced to copper 1 ions, Cu+. Now reduction is a gain of electrons. So when a Cu2+ ion gains an electron from the reducing sugar, it becomes a copper 1+, copper plus, or copper 1 ion in the presence of an alkaline sodium hydroxide, and this forms copper oxide. So copper oxide is an insoluble substance. It precipitates out of the solution as particles of a brick red color, a brick red precipitate. So a solution containing equal quantities of a food solution, let's say you have a food and you want to test whether there are reducing sugars in it, you make a solution by dissolving that food into a solution. You then mix that with an equal quantity of Benedict's solution, and you place that mixture in a boiling tube and then heat that boiling tube in a water bath, as is being shown here, for several minutes. Now, if you see the solution change color to this brick red precipitate, this indicates the presence of a reducing sugar such as glucose. So the brick red color is the definitive result. Now, when a Benedict's test is performed on pure solutions of glucose and sucrose, we see the following results. So here on the right-hand side, we see the glucose result. Here we can see the sucrose result. So sucrose is a non-reducing sugar because it does not produce the brick red precipitate, whereas glucose is a reducing sugar. So this is how we test for reducing sugars. What about the other category of carbohydrates, complex carbohydrates or polysaccharides such as starch? Well, for this we use iodine solution. So iodine solution is water with iodine, I2, dissolved in it, and it has this kind of characteristic reddish brown color. So when we drop put droplets of iodine solution onto a substance that contains starch, it will turn blue-black, like this potato, for example. So this blue-black color develops when the iodine reacts with starch. So this can be used as a test for the presence of starch. So here's another question for you to answer. This should pop up on your interface. Again, if it doesn't, then go ahead and answer in the webinar chat. What is the test that we use to detect the presence of proteins? What is it called, and how does it work? So I'm going to pause for a little while here and let you guys answer this question. All right, so well done. Looks like most of you answered that correctly across all 5 Zoom rooms. Well done. Excellent. So this is the Biuret test. So when we test for a protein, we use a chemical test called the Biuret test. So what we have here is sodium hydroxide solution and copper sulfate solution, when we mix these together and add them to a solution containing or a suspension containing a protein, you get this purple-violet colour. This is the definitive indication that the solution contained a protein. So we take a solution of the material to be tested, place it in a test tube, we add a little bit of sodium hydroxide, this clears the solution, and then a small amount of weak copper sulfate solution is then added down the side of the test tube. And then if you get this purple-violet colour, it means you have a protein present. If you don't, then there's no protein present. Well done. Okay, so here's another question for you to answer. So what is the chemical test that we can use? I guess it would be a physical test rather than a chemical test, because it involves a physical process rather than a chemical process. There's a clue for those of you who might struggle with this question. But how do we test for the presence of lipids, triglycerides, fats, and oils? So again, I'm going to pause here and let you have a go at this question. It should pop up on your interface. Again, if it doesn't for any reason, then go ahead and just type it in the type your response in the chat. And I'll pause here and let you guys respond. Okay, very good. Yes, many of you got this, most of you got this right. So yeah, it is the so-called emulsion test. There are various ways of testing for fats. I mean, there's also the paper test, but so testing for lipids, fats and oils, we use the emulsion test. Now, an emulsion is a mixture made from two immiscible liquids. Now, immiscible means two liquids that will not evenly mix with each other and then stay mixed. So if you take oil and water, for example, and try and mix them together by shaking, you will form a cloudy solution called an emulsion, which will eventually separate out into the two layers. So the way that we do this is we take the material to be tested, place it in a test tube, we add approximately 2 centimeters cubed of ethanol, and the tube is then shaken. The ethanol layer is then poured off into a second test tube, and then we add approximately 2 centimeters cubed of water to that ethanol. If we form a milky emulsion like this, then we know that there is a fat present. Now, the reason why the solution turns milky is because this region is an emulsion. It's not a solution. It is a suspension of fat droplets in the ethanol solution. An ethanol solution is mainly the ethanol solution has been mixed with water, and so this is water within which the lipid droplets are suspended, and this turns this region of the mixture cloudy like this. Okay, everyone, looks like we've come up to the end of the second hour here, so I'm going to pause once again for a 5-minute break. At the end of this 5-minute break, once again, you will see a poll pop up with the option to either for me to continue for an extra 30 minutes before going into the 30-minute Q&A session. There's been a lot of upvotes for 2 or 3 questions, and this is yeah, so it seems like across all 5 webinar rooms, most of you are voting for two or three specific questions for me to answer in the Q&A session. But once again, keep those questions coming. But take a 5-minute break, and then at the end you can vote as to whether or not you'd like me to continue for an extra half hour, either on exam questions or the next topics. So I'll see you in about 5 minutes time. Okay, everyone, we are back after the 5-minute break. So let's take a look at what you've all voted for me to do. Okay, it's fairly unanimous. It's about 80% or so have voted for me to go into the Q&A session now, so that's fine. If there are any other things that you'd like to ask me about, please do make use of the either the live chat on the private resources page on my website for this webinar. Again, you can find the link to that on the first page of your exam style questions pack. The mark scheme for this will also be uploaded to that same page, but please do ask any further questions that you have on that page. I will be responding all the way through to the end of June. Also, you have the ability to send me a private message there if you prefer to ask the question privately, but it would be nice for you all to get a discussion going around some of these topics. So let's take a look at what you've all voted for in terms of Q&A questions. So okay, so it seems like seems like the one question that has received the highest number of votes, or variations of this question have received the highest number of votes in almost all 5 of the Zoom webinar rooms, is it's concerning transpiration and the transport of substances in plants and how that relates to organization. The question is worded quite slightly differently here and there, but it seems to be what you're asking me about. So let's go into that. Now, again, all of the images of these slides for this entire PowerPoint will be uploaded, again, to that private resources page for those of you in attendance. If you're watching this video as a recording elsewhere, unfortunately, you won't have access to that. But if you're watching this on YouTube, you can go to the college vlog on my website, where I will be posting the interactive versions of these videos, which contain embedded pop-up questions and bookmarks and various other interactive features. So I would encourage you to go there if you're watching this on YouTube, because at least the videos on my website will be interactive. All right, so let's have a look at transpiration and the factors that affect transpiration in plant tissues, seeing as that's the question that most of you would like me to cover. So we've talked a lot about animal tissues. Now, like animals, plants have levels of organization based on tissues, organs, and organ systems. So there are 6 principal types of plant tissues, in the same way that there are 6 principal types of animal tissues. There is epidermis tissue, which is an outer protective layer on the surfaces of organs such as leaves. There are photosynthetic tissues, which contain cells which have large numbers of chloroplasts for the purposes of photosynthesis. There's also vascular tissue. Now, this is what we're going to be focusing in on, seeing as that's what you're asking about. Now, the vascular tissue, the word vascular, has the same meaning here as it does in animals, in the cardiovascular system. The cardiovascular system is made up of the heart, which is what the prefix cardio refers to. Now, vascular refers to tubes. So in the case of animals in the cardiovascular system, those are the arteries, the arterioles, the capillaries, the venules, and the veins. In the case of plant vascular tissues, these are the tissues composed of tubes through which you have mass transport of substances in the plant. So these are the xylem, which transports water and dissolved minerals, and the phloem, which transports sugar solutions and other assimilants. There's also meristem, which is the growth tissues where you have rapid cell division and differentiation from plant stem cells. Now, we covered that the meristem tissue in the previous webinar of this series on cell biology. And you also have packing tissue, which helps to bulk up the stem and the roots of the plant. Now, the final type of plant tissues are strengthening tissues, which have cells with thickened cell walls to provide strength and support for plants. So I'm not going to go into the epidermis and the other tissues here in too much depth. You can go ahead and have a look at those on the slides. I'm going to skip through these slides and go straight to the vascular tissues here. So vascular tissue, xylem and phloem tissues, this is the main transport system of the flowering plant. So a system of tubes called xylem vessels, which are coloured blue in this diagram here, transport water and dissolved mineral ions from the roots, where they're absorbed from the soil, up the stem and to the leaves. Now, the direction of flow in the xylem tissue is unidirectional. It goes from the roots to the leaves. So the phloem tissue, which is coloured red, is again a system of tubes through which you have the transport of sugar solutions. Now, the sugars are being made by photosynthesis in the leaves, but the sugars are needed by the growing parts of the plants and all of the other parts of the plants, because after all, sugars are required for photosynthesis. And so, unlike the xylem, the sugars in the phloem need to be transported bidirectionally, both up the plant and down the plant, because all of the different regions of the plant, particularly the regions which are growing most rapidly in the tips of the shoots and the tips of the roots, all of these regions of the plant require sugars to be delivered at a relatively high rate. And so the transport of sugars and sugar solutions in the phloem vessels goes in both directions here. Okay. So let's move on here. So meristem tissue, we mentioned before, so I'm going to skip past this very quickly and go to the actual transport in plants. So transpiration refers specifically to the movement of water. So we're going to be focusing on that. Now, much of the water in which plants take in from the soil is not used in photosynthesis. Now, again, think about the reaction, the equation for photosynthesis. Let me just quickly remind you of that. Okay, so photosynthesis is described using the word equation, where you have water combining chemically with carbon dioxide, and this forms glucose, a monosaccharide, and oxygen, molecular oxygen. Now, the water is H2O, carbon dioxide is CO2, glucose has the formula C6H12O6, and molecular oxygen is, of course, O2. To balance this equation, we need 6 molecules of water combining with 6 molecules of carbon dioxide to form 1 molecule of glucose with the production of 6 molecules of oxygen. Now, if we take a look at this equation, this means if this is what's happening in the photosynthetic tissues, which are found predominantly in organs such as the leaf, it means that water needs to be continually transported to these for the purposes of photosynthesis. But in fact, most of the water, a lot of the water absorbed by the roots from the soil, it is needed for photosynthesis, but much of it simply evaporates from the surfaces of the leaves. This loss of water by evaporation is called transpiration. Now, water is incompressible. It's an incompressible liquid, but it's also, the water molecules are cohesive. So if you have a tube of water within which, or a tube within which you have water molecules in the liquid state, those water molecules attract each other. So if water molecules are being lost from this end by evaporation, they pull on the water molecules beneath them. And so this causes the evaporation, the loss of water by evaporation from the leaves, causes a pull, which pulls water up from the roots. Now, this is a highly simplified description of what's going on here, but water lost by transpiration is replaced by a continuous column of water, which again carries dissolved mineral ions, which the plant also needs, such as nitrates and things like that. This causes a continuous flow of water to move up, and this pull, this transpiration pull as it's described, is enough to cause water to move up the trunks of even extremely tall trees against the force of gravity. So if you imagine a Californian redwood, which may be extremely tall, and you imagine just how large the trunks of those trees are, and think about the sheer amount of water that needs to move upwards. Water is actually quite heavy. When you imagine what a bucket of water weighs, imagine how much water is moving under the against the influence of gravity up the plant in that direction. Well, all of that, that force to overcome the weight of the water is provided by this mechanism where water lost from the leaves by evaporation from the stomata of the leaves causes this pull on the water molecules beneath, which in turn causes more water molecules to enter and move upwards. So this flow of water, this flow of this column of water is referred to as the transpiration stream. So let's take a look at the structure of the roots, because this is where the water is being absorbed. So roots, so this is a photograph of a root of a plant. You can see here that they're covered in many fine extensions which are called root hairs. Now, the root hairs are modified epidermal cells which create a very large surface area for the uptake of water. And so again, if you're having water enter these root hair cells, the larger the surface area that you can provide, the more rapid the absorption of water and mineral ions. Now, the structure of leaves is related to their function. Leaves are the photosynthetic organs of the plants. So, now, in order to fully explain the loss of water by transpiration from the leaves, we need to have a look at the structure of those leaves. Now, the water is lost through tiny openings called stomata, which are found mostly on the undersides of the leaves. So if we look at the underside of this leaf under a microscope, we would see something like this. So here we can see the epidermal cells, and you see the stomata, which are these openings which are controlled by guard cells. So this is one stoma. Stoma is the singular, stomata is the plural, and each stoma has a pair of guard cells which control the opening and closing. Now, the stoma, the function of them is to allow the carbon dioxide needed for photosynthesis. Again, to remind you, the cells in the leaf require carbon dioxide to enter them, and so the carbon dioxide needs to enter from the external atmosphere along with water from the vascular system, from the xylem. This is all delivered to the photosynthetic tissues within the leaf. The stoma will open to allow carbon dioxide to diffuse in from the external atmosphere, but also they open to allow the excess oxygen to dissolve out of the leaf, because oxygen builds up in the leaf. Some of it is used for respiration, but some of it the excess dissolves out via the stomata. Epidermis of that tulip leaf showing the stomata. This is at a magnification of 400 times. Okay, so here we can see a stoma. Now, the way that the guard cells open and close, they have a thick inelastic inner wall and they have a more thin elastic outer wall, and this allows them to open and close and produce the pore, the hole which allows the exchange of gases, carbon dioxide in for oxygen out. But these stoma also provide holes through which water can be lost by evaporation from the inside of the leaf to the external atmosphere. Now, this is a scanning electron micrograph showing a single stoma. Now, in the opening, you can actually see one of the cells within the leaf in the spongy mesophyll layer beneath the stoma here. So, guard cells take in water by osmosis from neighbouring epidermal cells. Now, this causes them to swell up and become turgid. Now, in the turgid state, they bend, so a gap develops between the guard cells. Now, the bending is brought about because the inner wall of the guard cell is thicker than the elastic outer wall. So, when the stomata are open, water vapour can evaporate from the inside of the leaf to the external atmosphere, and this is the process by which we call transpiration. So as the plant starts to lose more water than it's taking up from the soil, the stomata will close to prevent excessive water loss, and this prevents the leaves from wilting and drying out. The stomata close when water is drawn out of the guard cells by osmosis and then enters the neighbouring epidermal cells around them. So this causes the guard cells to straighten out like this and close the stomatal pore. Now, even on a day that's very hot and dry, the stomata do still have to open somewhat to allow for the exchange of carbon dioxide and oxygen with the atmosphere, and so you do still get transpiration taking place. Now, in very hot, dry weather, there is a danger that the plant can lose water by transpiration faster than it can be transported from the soil. Now, in these conditions, a plant will wilt because its cells have lost so much water that they no longer have that turgidity which keeps the plant upright. So here we see what happens to a plant when the stomata are fully turgid and it has sufficient water, and then this is the wilted plant which is desiccated, it's dried out because of excessive transpiration. So the situation is reversible if the wilting has not been going on for too long and the water is supplied again to the plant. So if you give this plant a bit of water, it will return to its former glory here. So there's actually quite a large amount of variation within the plant kingdom with regard to the number and the distribution of stomata in the epidermis of the leaves. Now, in general, the greater the frequency of stomata per unit of area of the leaf, the greater the rate of transpiration. And again, this stands to reason. If you have more stomata per unit of area, you will have a greater rate of water loss through those stomata. Now, in addition to the frequency of the stomata, their distribution, where they are found, is an important factor for determining transpiration rates. So, for example, plants that inhabit harsh, dry environments have stomata located in sunken pits to prevent them from excessive transpiration rates. So, such plants are referred to as xerophytes. So, let me quickly go on to the next question here. Okay, so I'm just trying to take account of the variations in that first question that have been asked. There's actually quite a few of them. The next question concerns drugs, specifically the drugs smoking and alcohol. Some of you are asking about other illegal drugs. All right, so let's quickly take a look at smoking, which seems to be the one which has attained the most votes here. Let me just find the relevant part of my PowerPoint here. And then we'll move on to smoking. So it seems like most of the questions concerning smoking are about cancer and how smoking causes cancer. So let's take a look at smoking now. So smoking releases a lot of carcinogenic chemicals. Now, cancer is the abnormal and abnormally rapid and uncontrolled division of cells to form tumours, which are undifferentiated cell masses, which can cause damage either by pressing on surrounding structures or by invading other tissues, what we call metastasis. Now, the chemicals present in cigarette smoke, there are many, many chemicals. These are just some of them. Many of these are carcinogenic. There are over somewhere between 400 and 600, depending on who you ask, different chemicals released by the burning of by the combustion that occurs in cigarettes and tobacco. Many of these are carcinogenic. So you even actually find radioactive isotopes like polonium-210 and cadmium. And certain isotopes of cadmium. Polonium-210 in particular is radioactive and does release alpha and beta radiation, which can cause ionization of cells, which can in turn cause cancer. Now, the reason why cigarettes contain polonium-210 is because many tobacco plants, commercially farmed tobacco plants, are farmed using inorganic fertilizers. Those inorganic fertilizers are mined from rocks which themselves contain high quantities of these and other radioactive isotopes, and so the plants they bioaccumulate those radioactive isotopes, and then when they are processed and dried and cured to make the tobacco, and the tobacco is smoked, those particles go into your lungs and start to emit ionizing radiation, which can then cause cancer. So very, very bad stuff. Cancer. So the way that this cancer is caused, not just lung cancer, but other cancers. Again, cancer refers to the production of tumours. And tumours are regions of abnormally rapidly dividing and uncontrollably dividing and differentiating cells. So let's finish off by looking at tumours in cancer then. So a tumour, or a neoplasm, is a tissue composed of cells that have grown abnormally. In normal tissue, the process by which cells divide to produce new cells, mitosis in somatic tissues, as we saw in the previous webinar on cell biology, mitosis is a highly controlled process in ordinary healthy tissues. So what you don't want in skin tissue, for example, is for new skin cells to be produced too rapidly. What you want is new skin cells to be produced at the right rate to replace earlier dead skin cells. And it's the same thing in other tissues. Different tissues have different turnover rates, and so the cells that produce the new cells produce them by mitosis. The rate of mitosis is very tightly regulated and controlled by genetic and other factors. Now, when those regulatory processes, when those control systems, which control the rate of cell division, break down in some way, the cells can divide uncontrollably and form these abnormal masses called tumours. Now, tumours are classified as either being benign or malignant. It's malignant tumours which are technically classified as cancers. Now, benign tumours they remain relatively localized. So benign tumours are abnormal cell masses, but they don't spread to other regions of the body. They just grow in one place. Now, the word benign is kind of misleading because benign in ordinary English means kind of, well, not malicious, kind of friendly in a way. But benign in this context doesn't necessarily mean non-harmful. Benign tumours, they do stay in one place. They grow as localized discrete masses. Now, they're rarely fatal, but they can be quite damaging because as they grow larger, they can interfere with the normal function of surrounding structures. They can displace adjacent organs, blood vessels, and nerves, and usually are treated by surgical removal because, again, they are quite well demarcated. The edges of a benign tumour are very clear and well distinguished from the surrounding healthy tissue, so they can be surgically excised relatively easily. However, malignant tumours, which refers to cancer, these display very different characteristics to benign tumours. Now, the most notable characteristic of malignant tumours is the fact that they can invade surrounding tissues and spread to other locations of the body where they can form new tumours. So this is really the more serious form. This leads to the more serious forms of cancer. So cells of malignant tumours. So what you're seeing here is a kidney which has a tumour in it. So here you can see the normal healthy kidney tissue, and here you can see the very clearly very different malignant tissue of the tumour here. So cells of malignant tumours have specific abnormal characteristics other than just uncontrolled cell division. First of all, the cells are undifferentiated. They are unspecialised. So you see here the normal healthy kidney tissue, like any tissue, has its unique appearance because the cells are uniquely and uniformly specialized. But you see the messy kind of nondescript appearance of the tumor tissue. This is because these are unspecialized cells. Now, the other characteristic of malignant tumours is that the cells of malignant tumours are not very cohesive. They tend to break away from each other and can go off into blood vessels where they can be carried to other parts of the body where they then divide uncontrollably to produce more tumours. So the cells can readily separate from each other. The cells of a malignant tumour can separate from each other and spread to form secondary tumours. Now, this is the process that is described as metastasis. Now, cancer cells can cause considerable damage, and they also compete with normal healthy cells for the available nutrients as well. So let's finish off by answering the major component of this question that has been asked in various forms. How do cancer cells spread? So normal cells, here we have normal cells in a tissue. Here we have a capillary running through the tissue here. So normal cells have mitosis happening at a controlled rate. But if you have a mutation in some of these cells, which causes a breakdown in the processes that normally regulate mitosis, they can start to divide uncontrollably and extremely rapidly, and these then form a tumor. Now again, cancer cells don't adhere to each other as well as the cells of healthy tissues, so these cells can break away and migrate into surrounding blood vessels, into surrounding lymphatic vessels, and then get carried around to other parts of the body where they can then multiply and form new secondary tumours, and this is referred to as metastasis. So, factors that contribute to cancer development, obviously smoking. So smoking is one of those things that actually contributes to, as a risk factor to so many diseases It's just, it's mind-boggling. So the carcinogens, the chemicals which can cause healthy cells to mutate, to become cancerous, there are many of them in cigarette smoke. So obviously smoking, whether it's direct smoking of that person themselves or living with a smoker and inhaling lots of passive smoke, this is a key risk factor to not just lung cancer, but many other forms of cancer as well. There are also carcinogens, chemicals in the environment which outside of just smoking, which can also cause cancer. So environmental chemicals and toxins can also be carcinogenic. There's also radiation, nuclear radiation, as well as ultraviolet radiation and X-rays. Ultraviolet and X-rays are electromagnetic waves which don't originate from nuclear materials per se, but they are found, for example, going out into the sun. Sunlight has strong sunlight has a high degree of ultraviolet. Ultraviolet and X-rays, these are at the high energy, high frequency end of the electromagnetic spectrum. They carry a lot of energy. So when these waves get absorbed by your cells, particularly by the DNA of your cells, they can cause the DNA to be damaged, and this can then lead to the mutations that cause cancer. Diet and alcohol is found to be a key factor in cancer as well, as are genetic factors. Some people are genetically more predisposed to developing cancer as well. There are also many viruses that have been associated with cancer and a higher risk of cancer as well. So let me finish off by the second component of this same question, which was about alcohol here. So let me just quickly find that section of my PowerPoint here. So alcohol obviously is a recreational drug. It is considered to be, the consumption of alcohol is considered to be socially acceptable for many people, but there are health risks. So a standard unit of alcohol has been established to help people control their alcohol consumption and protect, protect their health. So half a pint of medium strength beer, a single sherry, a single spirit measure, or a single glass of wine, all of these are described as containing 1 unit of alcohol. Now, there are recommended daily and weekly allowances for the units of alcohol. So how much is too much then? Well, the recommended maximum weekly allowances for females are 14 units and for males are 21 units. So why the difference? Well, females have a lower average body mass than males, and so and also they have a smaller liver. Now, the liver contains an enzyme that we saw earlier, alcohol dehydrogenase. Now, this is the enzyme which is responsible for, among other things, breaking down alcohols from your bloodstream. Now, if females have smaller livers, they have a lower capacity to break down alcohol, and consequently, the maximum recommended weekly allowance for females is lower than that of males. So let's have a look at short-term effects first. So drinking alcohol has a number of short-term effects. Initially, the drinker feels more relaxed, often becoming more talkative and less socially inhibited, but there are many more side effects. So initially, they'll start to feel relaxed, positively becoming more talkative. If they continue drinking, they'll become confused, unable to think clearly. After several more drinks, their coordination and movement will be affected because the alcohol is affecting the cerebellum, the part of the brain that's shaped like a cauliflower near the back, which controls and coordinates movement, and also results in slurred speech and an inability to walk without staggering. If they continue to drink beyond that point, their basic bodily functions will be affected, for example, breathing. Now, this can be very dangerous because it can result in them becoming unconscious, slipping into a coma, or in some rare cases, even resulting in death. Overdoses of alcohol can be fatal. So, let's look at long-term effects and finish off on that. Now, small quantities of alcohol are broken down rather easily by the body because, again, your liver contains the enzymes necessary to do that. However, when consumed in large amounts for long periods of time, chronically, alcohol can seriously damage the body. Now, the first place that gets damaged, among other places, is the liver. So, many serious effects, but alcoholism, chronic alcoholism, affects so much more than just the liver. So let's take a look at some of the effects. I'm just going to turn all of these on here. So alcoholic encephalopathy is a symptom of chronic alcoholism where the brain itself becomes severely damaged. You also have an increased risk of osteoporosis. The bones become more fragile and more likely to break. And obviously, if the person's a chronic alcoholic, they're more likely to fall over and therefore break those weaker bones. We also have liver cirrhosis, the loss of function of the liver. Now, the liver is the only organ of the body which is able to regenerate itself, but chronic alcoholism causes the liver to become fibrosed and loses its ability to regenerate itself, and the accumulation of the damage to the liver cannot be reversed until eventually this can become fatal. Alcoholism can also cause stomach ulcers. It can, again, we mentioned that alcoholism and alcohol intake is another one of these risk factors to various cancers, and of course it can also lead to weight gain and cause obesity with the associated problems that brings. Alcohol can also be addictive if taken in large quantities over long periods of time. Alcoholism is alcohol addiction and can lead to psychological symptoms as well as the physical ones, such as depression, severe anxiety, feelings of hopelessness and helplessness. So alcoholics drink to relieve these symptoms, which in turn just exacerbates the symptoms even further. Now, government statistics suggest that up to 9.7% of the population of the UK could be classified as being alcoholics. Now, although no genes have been identified which are linked to alcoholism definitively, there is considerable evidence to support the idea that there could be a genetic predisposition to alcoholism and other forms of addictive behaviors. Now, alcoholism is very, very difficult to overcome alone and can result, for the reasons already described, in suicidal thoughts and tendencies. But with support from organizations and from family, many people can break their addiction. Okay, everyone, seems like we've come to the end of the Q&A session. Thank you for those questions, and please do keep those questions coming in the on the live chat on the private webinar resources page that you will have access to as attendees. You can also send me private questions via the private messaging function on that page. I will upload this video as a interactive video with embedded questions and bookmarks, so please look out for that. If you are watching this as a YouTube recording, you can also find the interactive video on my website on the college vlog page. Just navigate to my website. Let me just bring it up here. And you will find this link over here. So I've already uploaded some of the webinar videos that I've edited to that page. You can find them here. So here's one on, let's see here. So here's one on structure and bonding, electricity on energy. You'll notice they all have these bookmarks. These little circles on the webinar video timelines illustrate where these questions will pop up, which will test your understanding. There are various types of questions in these. There are statements, there are multiple choice, there are single choice, there are calculation questions. So please do make use of these. All right, you're all very welcome. Thank you very much for attending. I hope that was all useful to you, and I will see you in the next one.